So the RAIO test is a simulation counter, measures radioactivity based on radioactive isotope of iodine, that is iodine 123, and the uptake varies greatly by the iodine status. That is indigenous. If you are taking indigenous diet, the normal uptake is 10% versus 90%. In presence of amiodarone, contrast-study, topical betadine, etc., this might change. So the elevated RAIU with hyperthyroid symptoms causes waves of toxic goiter. If your RAIU levels are low, then that could include diseases like thyroiditis, hormone ingestion, excess iron intake in Graves' disease, and ectopic thyroid carcinoma. So let's see what are the anti-thyroid agents that are present. So the anti-thyroid agents are hormone antagonists that inhibits the hormone production and transport. So there are several mechanisms of action that are involved. One is the inhibition of iron transport. Second is the organification of iron, where iron is added to an organic compound. Third is the processing of the thyroglobulin. Fourth is the transport of hormones to the peripheral tissue. In this figure, you can see that there is a transport of iodine molecules from the circulation into the thyroid glands, where the iodine molecules undergo oxidation with the help of peroxidase enzymes, and the oxidized iodide is incorporated into thyroglobulin to form monoiodotyrosine and diiodotyrosine, even T3 and T4. So further proteolysis will release T4, T3 into the blood, which enters the peripheral tissues, and the biological active form is T3. So thiocyanate and tetrachloride are negative inhibitors of the transport. Iodides and thiamides are negative inhibitors of organification into oxidized iodine into thyroglobulin. Iodides inhibit the proteolysis. Radiocontrast media, beta blockers, corticosteroids, amiodarone, etc., inhibit the action of T3 in the peripheral tissues. So the antithyroid drugs include the following classification. That is the inhibitors of hormone synthesis. Example is carbimazole, methimazole, propylthiouracil. Inhibitors of hormone release, that is iodine, iodides of sodium and potassium, and other organic iodides. Destroying of the thyroid tissue by radioactive iodine. Inhibit ion trapping, ionic inhibitors. Example thiocyanate, perchlorates, nitrates, etc. Inhibition of hormone synthesis. So methimazole, propylthiouracil, these two are the major drugs that is used, particularly for the treatment of thyrotoxicosis. And the mechanism of action is where the drug inhibits thyroid hormone production by inhibiting thyroperoxidase required for intrathyroidal oxidation of iodine, inhibits the iodination of tyrosine by inhibiting coupling of monoiodotyrosine and diiodotyrosine to form T3 and T4, and PTU also inhibits, or propylthiouracil also inhibits peripheral conversion of T4 to T3 by inhibiting the deiodinase enzyme. So the control of thyrotoxicosis in both Graves' disease and toxic nodular goiter. Second is the inhibition of hormone release, where the iodide salts inhibit organification of iodine and hormone release. These salts also decrease the size and vascularity of the hyperplastic thyroid glands. The onset of action occurs rapidly within 2 to 7 days. This effect is transient, that is, the thyroid gland escapes from iodide block after several weeks of treatment. So drugs include Lugol's solution and saturated solution of potassium iodide. So next is the use of iodinated contrast media, also called as IPODATE. It suppresses the conversion of T4 to T3 by inhibiting thyroid deiodinase. IPODATE has also proven useful in reducing T3 concentrations in thyrotoxicosis, or also called as thyroid storm. The third is destroying a thyroid tissue by radioactive iodine, which is administered as sodium salt. The iodine 131 emits X-rays as well as beta particles, and it can be concentrated and incorporated in choroids, thereby emitting radiation from within the follicles. So as a result, the follicular cells undergo pignosis and necrosis, followed by fibrosis with a large dose, leading to massive tissue damage. Next is ionic inhibitors, that is, presence of certain monovalent anions that inhibit iodide trapping by NIS, similar ionic size as compared to 1, and the T4 T3 synthesis is also inhibited. Toxic, the drugs used are usually toxic, so it is not usually preferred. Examples include thiocyanates and perfluorines. Other drug treatment options include beta blockers, example propranolol. It is used to rapidly alleviate toxicosis due to sympathetic overactivity, palpitation, tremor, nervousness, and sweating. In addition, they reduce peripheral conversion of T3 and T4. The beta blockers can also be used in hyperthyroidism when you are awaiting a response to propylthiouracil for preoperative preparation, including thyroidectomy and thyrotoxic crisis.